Despite the presence of better technologies for biomedical research, experimenting on animals is still the norm. Why is that? Keep listening and hear from a former animal researcher about the severely broken system that is preclinical research and what is needed to change it and get safe and effective medicines in the hands of healthcare providers more quickly to help treat you. Welcome to Puppies, Pandemics, and Public Health. I am Dr. Johnny Lieberman. If you've ever wondered how your everyday choices, including what you eat, what you buy, what you wear, and the medicines you take, impact your health, the health of your family, animals, and the planet, you are in the right place. Let's get to it. Welcome to another episode of Puppies, Pandemics, and Public Health. Today, I'm pleased to welcome Dr. Emily Trunnell. Dr. Trunnell graduated magna cum laude from the University of Georgia with a degree in nutrition science and also earned a doctorate in neuroscience, also from the University of Georgia in 2016. Dr. Trunnell is currently the Director of Science Advancement and Outreach, a division of People for the Ethical Treatment of Animals, PETA, which works to modernize science policy, scientific publishing practices, and university scientific education and training to support the transition to animal-free research. Dr. Trunnell, thank you so much for being on the show today.
Thank you for having me.
We had spoken before, and I seem to recall that you actually have been in a lab where animals were experimented on for research purposes. Is that correct?
That's right. I did a lot of the experimenting myself. So this was during my graduate training when I was doing my PhD in neuroscience. We used mice and rats, predominantly rats. And it was that experience that led me to come work for PETA, to be honest. In the beginning, knew a bit about animal rights. I ate vegan and I loved animals. And I went into this thinking, this is just how we do science. And I want to be a scientist. I was kind of excited to be around animals all day. Mice and rats get a bad rap, but they're remarkable animals. They each have their own personalities. And over the 4 years, I really started to question, is this worth it? I saw things happen in the lab, accidents, animal suffering.
Can you give an example of one thing that you saw that made you kind of question what you were doing and if it was worth it?
Yeah, so we were doing this one study where we had, I think it was around 30 female rats, and they were older rats. They were retired breeders that we were using to study menopause. And they were each housed in their individual little shoebox cages because we were feeding them all different diets. And we were required to go in every day and we were doing the feeding. And at some point, somebody had put the rat's cage in the wrong way. And what that meant was on the cages, there's a hole in the back. And in the rack, the little spout comes through that hole where they can get water. And I don't know how many days this cage had been in backwards. So that rat had no access to water and she was completely dehydrated and emaciated. I'm surprised she wasn't dead when we found her, but we did have to euthanize her. And then some of the different rats in another study were in the same room as rats from other labs. So their cage rats were in the same room as other studies. In this other study, they were studying alcohol and alcohol's effects on the brain. And so they would get these rats drunk. They would inject them with alcohol. And I was doing my own thing and then looking over at the cages after the other people had left, and these rats just looked miserable. But it was kind of presented like, this is funny, we're getting rats drunk. And I even took some pictures of them because they had no power over whether or not we could all go to the bar after a long day in the lab and choose to do whatever we like, but this was being forced upon them. And so things like that. And then really a lot of it was the fact that the experiments I was designing and doing weren't really to serve a greater purpose for knowledge or medicine or health. They were really just to get me my dissertation. We just designed studies that kind of made sense, like thinking about a question we were asking. Nobody was looking at the big picture and saying these experiments need to be done. Are they going to tell us anything? It was just, you need 3 papers to make 3 chapters of your dissertation, and they all have to be different experiments. And I submitted the applications to our animal use committee, and they got approved, no problem. The system isn't set up to stop anybody from doing anything. You can do anything you want to an animal in a laboratory if it's in the name of science. And the more animals that I experimented on and even harmed and killed, that didn't add up for me.
Yeah, you bring up a few really good points. Thanks for sharing that, Emily. It goes beyond just a question of, do the animals suffer and how much, and the autonomy that they lose over their own bodies, like you said, the powerlessness. But all that suffering, it doesn't matter in the big picture. And for me, it makes it more egregious. It doesn't matter. For you, it's to get papers published, and it matters to get money, correct? Someone writes checks for these experiments, and it sounds like the use committee or oversight committee signs off and says, yeah, this looks good, perhaps knowing that the chances that's going to contribute anything to human medicine is 0 or close to it. Yet the funding keeps coming in. Is that kind of an accurate assessment of our current system and one of the major flaws in it?
Absolutely. So they're called institutional animal care and use committees, or IACUCs.
Would you explain that for the audience? There's a kind of mandate for that.
Yeah, the Institutional Animal Care and Use Committee, or the IACUC, is a group of people that's at every institution that uses animals. It's required in the Federal Animal Welfare Act. And they essentially review and approve every study, every experiment on animals that someone in that institution does. So this is universities and foundations and things like that. They just have to make sure that they're checking the boxes of the Animal Welfare Act, which are very minimal. So you would expect this ethics committee to ask, do you really need to use animals for this? The only thing that they have to make sure is that the experimenter considered it. They thought about it. They don't have to have any proof that there is a different way to do the study that doesn't use animals. But they also have to make sure that the animals are being fed enough, that they're supposed to check whether or not an animal is given pain relief if something is going to be painful or distressing, but with the caveat that if it goes against the scientific aims of the experiment, say the person wanting to do this says, well, we can't give pain relief in this study because that pain medication will mess with the biological process we want to study. So it would confound the experiment. Okay, no problem, as long as you give that justification. And the thing is, when there are whistleblower reports or undercover investigations in laboratories that show the horrific things happening to animals, or somebody releases like a USDA inspection and they are getting a little bit of flack, the university says, no, everything's okay. We have the IACUC. It's IACUC approved. This is a very rigorous committee that judges the experiment and makes sure everything's up to code. But they don't really do that. They check some minimal boxes, which can all be another one of them is animals aren't supposed to be put through multiple invasive surgeries without justification. So the scientist writes the justification. Well, this is why we need to do this. That's never questioned. They just need to check the box that, okay, they gave us something to say why. So it's really window dressing. So that oversight is not oversight at all. And another thing about them is that the composition of these committees. So the law is that they have to contain a chair, someone who's a scientist. They have to have one veterinarian, and they have to have somebody who is not a scientist, but they work at the university. And then they have to have supposedly a member of the public, which when Congress set this up, they were like, oh, the member of the public who's not affiliated with the institution, who's not a scientist, kind of act in the role of the public and think about things from what we would want from our own morality. But in a lot of cases, those are just a few of the mandated people. Most of the time, the IACUC is full of other animal experimenters. So they're really policing themselves. So fox guarding the henhouse. And we've even uncovered, PETA had a lawsuit recently against the University of Washington to reveal who was on their IACUC, because we had a suspicion, and we were right, that the person who was supposed to be the community member, not a scientist, supposed to have no vested interest in animal experimentation, was like the director of a pro-animal experimentation lobby group, which doesn't fit what Congress set that role to be.
A couple of really important take-homes is it's really a self-regulatory system is what this is, which is not the intent of it. I don't think was for it to be so much that way, but that's what it's morphed into. Anecdotally, from speaking with others in my area who are involved in this space, what these facilities tend to do, if they are complying with the law in terms of having one non-affiliated member, while there's a minimum of, I think it's 3 members with the AWA to have on the committee, they might have 15. And so 14 of them are going to have their paycheck signed from the university or the facility that's getting the money. And guess what their incentive is going to be to sign off on these procedures? It's really going to be more like a rubber stamp. And then someone standing up and say, hey, do we really need to not give these animals anesthesia? Is that really going to affect the results of this? No. Self-regulation. Do you think that works in other areas that are important? Does it work in the airline industry? How comfortable would you feel if a major airline was responsible for overseeing its own safety, for example? Or if doctors were in charge of self-regulating what we have to do for continuing education and maintaining our practice standards? It's a failed system.
You're absolutely right. And I think if you're that one non-affiliated community member against 14 scientists who are all saying, no, this checks out, this is good, that's completely intimidating. And even if you were to vote no, that this shouldn't be done, you're going to be overruled. But most of the time, we've heard from people who have served in this role that they felt like they had absolutely no voice. They were shut down. So it's not set up in a way that leads to improved animal welfare.
Horribly broken system. I think a lot of the public, if they even know about them, I think there's an impression that's given that, oh, well, there's an ethics committee mandated by the law at every facility, making sure these animals are treated ethically before signing off on experiments. That's nowhere near the truth. No, it's a rubber stamp to check a box, like you said, and move along in the process. One other point before I let you continue is you mentioned the Animal Welfare Act as it is enforced on facilities. That doesn't even cover an overwhelming majority of research animals, correct?
That's right. So the Animal Welfare Act excludes somewhere around 95 to 99% of the animals that are actually being used in research and testing. It excludes mice, rats, birds, fish, any animal that's being used for food production. And these are the animals that are, mice especially, are about 95% of the animals used, and they have zero federal protection under the law. And the only reason, obviously, so this is how the Animal Welfare Act defines animal, as excluding all these animals, which I always say that's not very scientific because everyone knows they're clearly animals. But it's convenience because there are so many mice and rats and fish. They just don't want to have to count them all. There's no accurate number for how many animals are used in experimentation and testing in the US. Other countries, they count mice and all these animals. They can give figures, but the US has never been able to give a solid number. The estimates are around 100 million animals per year, if you include missing.
100 million animals a year. That's a number that's hard to grasp, but it's so many animals. What's the U.S. population? It's like 330 million? Is that a ballpark?
Oh, goodness. I can Google it.
Well, give us an answer right now. I think
it is 342 million.
All right. So I wasn't too far off. So 342 million people living in the U.S. We have 100 million animals being experimented on. 1/3 of the U.S. population in sentient beings, an estimate. I speculate it's probably more than that. But that's a massive amount of animals being subjected to painful experiments, in some cases, no oversight for their welfare. And for what? What are we getting out of it that contributes to human medicine?
Not a lot. I'll answer that, but I also wanted to add, so the Animal Welfare Act obviously is the federal law that governs the use of animals in this way, but there's also the PHS policy or the Public Health Service policy, and that's supposed to provide some additional oversight to any animals used in experiments that are funded by any agency under the Department of Health and Human Services. So predominantly NIH, but some other agencies as well, or National Institutes of Health. And I won't keep going on about this, but just so mice are covered in PHS policy, but that agency, the Office of Laboratory Animal Welfare at NIH, doesn't do any laboratory inspections. They never set foot in a laboratory, and they also don't require animals to be counted. When there's a problem at a laboratory, the university will write to the Office of Laboratory Animal Welfare and say, oh, we had this problem, but we fixed it. Don't worry about it now. They're supposed to self-report these things. And the Office of Laboratory Animal Welfare will write back, we acknowledge your problem. It sounds like you fixed it. Thumbs up. That's it. They just completely rely on letters sent back and forth, never set eyes on any animals. So it's obviously completely insufficient. But
So it's absolutely right. It's another mechanism that works through funding, whereas the AWA does not. It's self-regulatory. It's set up to fail.
Yeah, and there's no incentive. Each institution that gets NIH funding uses animals is supposed to have a public health service.
Sure.
The PHS assurance. If you're a university and you have faculty that want to do experiments on animals and NIH is willing to fund it, you're not going to say no to that. That is money coming into your university, and this is a whole another topic too, but universities get to skim a ton of money off of the top of federal grants. These
legally
overhead costs or indirect costs. So there's no incentive not to do completely useless animal experiments because you're getting funding. And the university doesn't even really have to use that money to pay for the experiments. They could use it just for administrative salaries or things like that.
It's great information. It's building this picture of a horribly broken animal research system. It's horribly broken.
And it's frustratingly complicated. It leads to a lot of confusion about what happens to animals in laboratories and what's allowed and what's not. But you asked, what are we doing all of this for? What are we getting from this? And 95% of the time, nothing. So mostly nothing. Animal experimentation has been kind of a standard in biomedical research for hundreds of years. And we're still using a lot of the same animal models and experiments that we were in the 50s. And the statistics about what that actually results in, because a lot of people think of this as a necessary evil. I don't like what happens to animals in laboratories. I don't want to see it, but this is how we do science. We have to do this to test medications or to find the cure for cancer. So when it comes to drug testing, for example, up until not that long ago, all drugs that went into human trials were required to undergo animal testing for safety and efficacy before going to trials. Up to 89% of research can't be reproduced from laboratory to laboratory. And in science, being able to reproduce an experiment is really a hallmark. Is this a true theory? And 89% of the time, that can't be done.
So just for the audience to clarify that this lack of reproducibility is a major problem. That means if one lab, for example, say in New York, does a study with rats and they say, oh, we've got an 80% success rate for this drug for curing leukemia. And a lab in, say, California takes an identical breed of rats, identical number, performs the same doses of testing, the same experiment, everything exactly the same, and they get a different result, right? Maybe they says, oh, it didn't do anything. It was the same as the placebo. So who do you believe? I could guarantee you this, the 80% is going to get published much more likely than the one that showed 0% success rate. And that leads to a whole nother problem with these statistics and reporting something that's a success. It's been estimated that for every publication that shows a successful treatment in the preclinical setting, there's about 4 which are available, which don't show that, which show the opposite effect. So very, very important, this lack of reproducibility, separate from the translatability actually, but it's lack of reproducibility. That's a major flaw that exists throughout animal research.
It does. And I mean, the reproducibility is even a few years ago, people started calling it the reproducibility crisis because it not only affects the animal experiments, but also it can affect human research and cell studies. But I always think about when animals are involved, there's an extra harm that's involved. You're not just wasting money on cells in a lab, you're harming an animal and taking a life. And I think that should have a bit more of a weight. But yeah, what you mentioned about these positive studies getting published much more often than the negative ones, that's a publication bias that's been talked about a lot. And it gives a false impression that experiments on animals are leading to good things. When you see the headlines, cancer in mice cured. Okay, we've cured cancer in mice 100 times or more. We're not going to find a cure for a human. These headlines give this false impression that, oh, the scientists out there experimenting on animals are doing fantastic things and these cures are right around the corner. And that you don't see all of that behind the scenes, the failed reproducibility. Yeah, so that's another huge problem.
Yeah, thanks for fleshing that out a little bit more. Huge problem. Well, I'll let you go. Where do you want to go to next?
Do you want me to go a little bit more on translatability?
It's separate from reproducibility, but yeah, I think that's very important. Mice aren't humans.
Right. We don't live with our heads an inch off of the ground. There are so many things about animals that are different from us, even though there are many things that are the same. I think that's so these species differences are extremely important in the way that we are doing science today. Because we're not back in the early 1900s, just kind of studying basic anatomy and cells. The way we're developing drugs now and the levels at which we're understanding human biology are extremely to the finest point. One of the things people say when they are explaining why we use mice, for example, in research is that, oh, well, humans and mice share 85% of our DNA. So there's so much in common. But what matters is how those genes are expressed. So you can think of like the keys on a piano, and with the same keys, you can play an infinite number of unique songs. And that's how, while we share 85% of the same genes, those genes are expressed and regulated in very different ways, which is why we don't look like mice, why dogs can't eat chocolate and we can. So many different things about our biology just doesn't compute. And this translatability, these species differences just compound when you have animals in such unnatural conditions. For example, in laboratories, mice are kept at extremely cold temperatures, and activates the stress response. They don't have natural circadian rhythms. So there's no dusk, there's no dawn. The lights are on, they're off. Experiments are often done on animals at times when they would naturally be sleeping if they were in the wild. And I don't really want to be woken up. If I'm woken up in the middle of the night for a behavioral test, I'm going to kind of behave a bit differently than if I did it if I was awake. And then, the diets, the noise, so many of the setting itself confounds experiments. And that's actually one of the things that reduces the reproducibility as well.
It's like it's set up to fail from the beginning when you have all that.
Exactly. And I mentioned 95% of new drugs fail in human clinical trials, even though they've passed animal tests. The numbers are even higher for some areas of research. 40 years of experimenting on monkeys and mice, and there's no HIV vaccine. Sepsis is another area where, really all of the treatments tried in mice have failed. So in some of these fields, it's more like 100%.
Sepsis is a very actively studied area, as I think in an area where I've practiced for over 20 years. I was in practice when a lot of the excitement was over vitamin C for sepsis and another drug called activated protein C, which I never read the animal data, but because that's not what I do as a practicing clinician, but it must have been solid to get it into clinical trials. And those drugs, the vitamin C and the other drug were busts. And that's because of the issues we're talking about. If you would have not known anything about it, the prediction would have been they're probably going to fail, knowing everything without having read any of the data. And that's what happens. We wasted maybe 10 years in preclinical work, probably billions of dollars, smart researchers' time, probably lots of publications that are out there, and to bring drugs to market, which have a high probability of failing despite showing success in the animal models. And that's a routine, what Emily is talking about. That's not the exception. It's the exception when something works in animals and then it works without the unintended side effects in humans.
Yeah, that is an exception. It's not the rule. A broken clock is right twice a day. People who advocate for using animals in research, they like to point out some of the successes. And they're good things, but it's not because we used animals that we got certain treatments or certain vaccines. Animals were part of the process, but they are not necessarily the reason. And they've always been included, unfortunately.
And I argue, and one of the ways I look at it is they're potentially slowing down progress as a lead in perhaps to what you're going to talk about next in an area which you're familiar with perhaps more than I, something like Alzheimer's disease, which affects so many people. And there's animal models for it, but they're terrible. It's been studied for decades. There have been promising therapies that appear to work with the understanding that, again, even what we call a surrogate, which I'm guessing is what's used in these models, because you can't give a mouse a mini mental status examination, or you can't give them, tell me what 12 minus 6 is. They're not going to tell you. So even the concept of it as a clinician, it's like, what are they even thinking? Of course it's going to fail. Not to mention, we don't know the underlying pathophysiology of why it happens in humans. We have an artificial created model, which researchers think is probably good, but it just, as a clinician who treats diseases, you have to understand the pathophysiology, why the disease develops. Sometimes you have treatments and you don't know exactly why it works, but as clinicians, we're leery about using those medications. So it does happen though. But in general, you need to understand how the disease happens And if you are going to use an animal model, make that same condition happen the same way, and then look at treatments. That's not what's being done. And so, again, not knowing anything, just learning what you learned on our episode so far, that shouldn't be surprising that after decades and billions and probably millions of animals sacrificed, we have a couple of treatments on the market for Alzheimer's, which don't cure it, don't reverse it. A couple of them slow the progress a little bit, but that's all we got. Successful in animals, again, poor translatability to humans. It's the rule, not the exception.
Yeah. And some of those new Alzheimer's drugs, I know I've read there's been concern about their safety as well, or in certain populations because of reading in the brain. There are so many mouse models of Alzheimer's disease. Mice don't naturally get Alzheimer's. So even what they're studying is something that looks like or has a few of the symptoms of Alzheimer's. But you're right, the cognitive tests for mice are very different than what we would do with humans. And it came out a few years ago, the basis for the amyloid hypothesis for Alzheimer's disease was based on a study in rats that was even fraudulent. Some of the data in the paper had been faked. So many resources misled because not only were they using rats, but they were also fraudsters potentially in what they were doing.
I didn't know that. So thanks for sharing. Do you want to share the amyloid model, how that connects to Alzheimer's in humans?
Sure. Maybe you correct me if I'm wrong, but it wasn't the area I studied. But essentially, these amyloid proteins build up and aggregate and kind of go haywire in the brain and disrupt the signaling of the brain, which disrupts memory.
Yeah, that's about how I understand it. I don't think it's even proven cause-effect relationship, but these abnormally folded proteins accumulate in the brain. They're seen in patients with Alzheimer's disease at greater incidences of the brains of patients who don't have Alzheimer's disease. Do they directly cause it? I don't think that's 100% known. And how or why do they develop? I'm pretty sure that's not known. So with those two questions unanswered, creating, like you said, a similar model in mice, no wonder they fail.
Yeah.
So yeah, there's a lot of work to be done. There may be better ways to do it.
This is not the 80s anymore. We have incredible technologies now that can replace the use of animals and generate data that's even better than what the animal experiments were generating. There's several categories of these. I call them non-animal methods. Some people call them new approach methods or new approach methodologies. I like the term non-animal methods because it's clear. I think, we want to emphasize that the goal of biomedical research for human health should be based in human biology, not other animals. Sometimes you'll see the acronym NAMs, which can be used to mean all those different names for them. But they're fantastic. I actually just gave a presentation last night to a student group at the University of Washington all about NAMs. And there was so many studies I wanted to put in there because the amount of work is just incredible. But broadly, they're in a few categories. So we have in vitro methods, and in vitro just literally means in a dish. So you may have seen cells in a little petri dish. That's not what we're working with anymore. We can grow cells in three-dimensional structures. We have models called organs on chips, and I actually have one. I can show you what they look like. This is an older model, but this is from a company named Emulate. Let's see if I can get it in the screen.
Yep.
And so it's a flexible thing. It's about the size of a small computer memory stick.
So what's on there? Tell us.
So it's flexible plastic, and then there's two channels. You'll see one, they're different colors. And so what they do is they take human cells. For example, you could take cells, and these can be cells that are collected from a skin sample from a patient, from a urine sample, from a saliva sample. And one great thing about it is you can use any human, or you can use a person who has a specific genetic disease or a chronic condition. So you take a sampling of their cells, and then scientists can program those cells to become different tissues in the body. So they can turn some of those cells into airway cells. So that might be in the blue channel. And then they can turn some of the other cells into blood vessel cells. And so that might be the other channel. So the cells are grown on these tiny little channels. And then you probably can't see it, but there are holes on either side of the channels, and that connects to a device that is like a vacuum pump. So it pulls and stretches the channels the same way that you'd see if you were breathing in air or blood was flowing through a vessel. And that physical movement helps the cells grow in a way that they would naturally grow in the human body. Now you have a system, this is the lung one's just an example, but you have the human airway and it's interfacing with a human blood vessel. And so you can run particulate like matter through the airway and see what gets into the blood vessels and how they react. You can run a drug through the blood vessels and see how it affects the cells in the airway. And this is a really simplistic version. You can have multiple tissue types, more than two. You can connect, say, a heart on a chip to a liver on a chip and study how molecules move through that system and react to drugs. Those are super. So there's a ton of great YouTube videos if you want to get really nerdy. But we have other ways that we can grow cells now that allow them to behave like they would in a human, and they retain that human biology. So in other ways, they're called organoids or spheroids, and they're like little clumps of cells that resemble a tissue. So we have brain organoids, liver organoids. Most of the tissues and organs in the body have been mimicked this way. It can connect those and form assembloids, by connecting, again, like a heart and a vascular organoid together, or cardiac cells and vascular cells, or you can grow regions of the cortex, so regions of the hypothalamus, different parts of the brain, and create a more functional human brain tissue. So these can also be used to really just study biological mechanisms or test drugs. So those are the cell-based models. We also can do incredible things with computers, especially now that the AI has entered the picture, that they can really predict how cells behave. And you can run experiments in a computer without ever having to touch an animal or a human or even cells in a dish. Some of these computer models are predicting, like if a chemical is toxic, they're out predicting what the animal experiments did.
Let's pause right there, Emily. These are both such exciting technologies. And I just want to reframe it a bit for the audience. Like the last one, for example, you were talking about lung. That was like we call alveolus on a chip, approximately, which is replicating in the human. The alveolus is the most distal part of the lung that interacts with blood. So you inhale stuff, whatever you inhale, it goes into the alveolus eventually, and a blood vessel takes it away to the rest of the body. So with this example, for example, you wouldn't have to expose dogs, for example, or to cigarette smoke and see if they get lung cancer. You could have the chemical introduced into this system and see what happens to it at that alveolus level. And if you connect it to liver, like you said, you have these, what do you call them when they're two organs connected again?
Assembloid.
Assembloid. So assembloid, right? You can see if it's going to the liver, is it causing liver damage? And it's real, it's human tissue. You don't have to extrapolate what happens in zebrafish or what happens in a mouse. Might that happen in humans? I don't know. Might, but it might not. But you've got human cells. And at the same time, as Emily was talking about, you can screen potentially thousands of molecules if you're looking for potential toxins and come up with, using AI, for example, in silico modeling, come up with a list of maybe 20, which are high probability or likely to be worthy of testing or cure, if you're looking at it from a cure side, and do the same thing. You can inhale it if you want. You could directly expose it to the liver. We have liver on chip models, right? You have a mini liver of human cells. Liver is one of the common organs that gets a lot of toxicity because it filters out drugs. That's kind of part of its job. So it's a common side effect of many medications, but also it's a target for many toxins that we get in the environment. So not only does this technology represent more human relevant way to do it, it's faster. It's faster. You don't have to wait 5 years or however exposing an animal to a toxin or trying it on the therapy and see what happens down the road. You find out pretty quick and you avoid the animal use. Yet it just makes so much more sense. I wanted to frame that on every level. So please, Emily, continue.
Yeah, you mentioned a liver on a chip. So there's a great study that I talk about a lot. And this is a few years old now. So things even improved since then. But there was a drug that came out in the 90s. It was an antibody drug. And they put it through animal testing. It was standard. No toxicity, no adverse effects detected in animals. It went into human clinical trials and patients started getting blood clots. And so the clinical trial had to be stopped. This company, I think it was Johnson & Johnson, they went back and partnered with Emulate, the company that made this organ on a chip that I showed. And they tested that same drug to see if in a liver, it was a blood vessel, but they've since done great liver stuff. So this is a blood vessel I'm talking about. They tested this drug that had caused blood clots in humans and the blood vessel on a chip with human cells, and they saw the clots. So this cell could have prevented this model using human biology could have predicted what the animal experiments could not. That's just a great example of how the species biology really matters. And now that we have these tools, we can make sure that what we're putting in our bodies is safer.
Wow. I didn't know about that story. I'm guessing you probably have more of those that you're aware of, which I feel like the dominant narrative, at least that I see, is that animal research is critical. And many researchers and many in the field don't want to give it up. They think because it's been the past, it's the best way going forward for the future. And stories like this, in my opinion, need to get out there and change this narrative that relying on animals is holding us back. It's giving us false information. It's giving us false hope and causing real world toxicity that we have the technology to avoid now.
Exactly. It's not correct that, if we get rid of animal testing, the drugs will not be safe in clinical trials. The way it is now, drugs having gone through animal tests pretty much only, and then going into human clinical trials or have gone through a very simple cell model, not these advanced ones. The first person who's exposed to that new drug is still the proverbial guinea pig. It has never been in realistically grown human environment yet. And the animal tests are so unpredictable. So these technologies really can, for safety especially, give us a more realistic picture of what that person's going to experience if there's going to be any side effects before they're the first person to be exposed to it.
I'm glad you brought that up. That's another great narrative, in my opinion, that's not out there. There's a general sense in the public and many people who are outside this space, which is most people, that, I'm happy to sign up for a clinical trial. It's safe in animals. Yeah, let's go. And as we talked about, the probability is that what we found in animals is not going to be what we find in humans. Yeah, I'm glad you brought that up. Human testing has to happen before it gets FDA approved to go for general use. What happens before that is what we're talking about now, the so-called preclinical testing. We have an outdated, non-translatable model, or we're talking about models which are actually based on human cells, which have a much better predictability.
Yeah, and just in 2025, the rules finally started changing. It was long overdue. So the rule, and it was a loose rule, but it was very much perceived as a rule, was that before a drug could go into humans, it had to go in two species of animals, typically a small animal like a mouse or a rat, and a larger animal, typically a dog, a pig, or a monkey. But now the FDA Congress passed the FDA Modernization Act, and the FDA is also putting out their own new roadmap that it says literally in their release, they want to make animal testing the exception rather than the norm. Similarly, other agencies in the US, like the NIH and the UK government last year, all announced similar roadmaps. And so the promise of these technologies is finally getting realized, and there are efforts to really replace the use of animals with them. They still
need a lot of work, and funding shifting can get us there for sure. I have no doubt that's happened in the past when government decides this technology is important for public safety and they throw money at it. Guess what? Things develop, things happen. We already have a substrate, so to speak, here for doing this. We've got it, and it can grow and expand and completely replace, if not completely replace, eventually. And I think that's the goal we're moving towards. Our government has recognized it. And you mentioned the statutory mandate for using animals before going to human trials. That came from 1938, the Federal Food, Drug, Cosmetic Act of 1938. Not quite at 100 years, but we're north of 80. So doesn't it make sense to modify that how much technology has changed since 1938? Yep, exactly. Isn't it past time to bring us up to date and not be stuck in the World War II era, 1930s, 1940s for biomedical testing?
Yes, it's long, long overdue. And we're hopeful that things are going to start changing. I think things just kind of peak together. We have these amazing technologies, and they're really advanced, and they're getting the recognition they deserve. And hopefully we'll start getting the funding they have deserved for a long time. The public sentiment, the percentage of people all over the world really who are against the use of animals in research and testing is larger than it's ever been. That number has, in people who oppose it, it's increasingly gone up. And then we have growing recognition of the inner lives of animals and how they experience the world. That's another side of science that is learning about animal sentience. And then, of course, there's different political wills. A lot of the political will, especially in the US over the last year, has been focused on waste and getting rid of government waste and reducing government spending. And honestly, the animal research, very easy, low-hanging fruit. It is extremely wasteful. People don't like it. And we have the technologies to replace it that can help us get medical breakthroughs a lot quicker. So all this will kind of came together in the last few years. And so I'm hopeful.
As am I. And we need to keep pushing forward, right? No letting up. We've got some momentum going. We have the mindset in some of our federal government, as you mentioned, to move forward. Talked about the FDA. And yes, efforts like yours and so many in this space, I am hopeful that we can keep things going in this direction.
Name as well.
What else would you like to share, Emily?
I just wanted to plug my work in PETA Science Advancement and Outreach Division. We work on science policy. We work on improving standards at journals, and we work on supporting people in graduate and undergraduate degrees and postdocs who want to be great scientists and want to avoid the use of animals. So working to change that landscape as well. We have a big policy roadmap called Research Modernization Now, which lays out the steps. They're very common sense steps to shifting away from an animal-based biomedical research paradigm to one that's human-based. Any listeners interested in all that, you can find us at scienceadvancement.org.
Thanks, Emily. Very happy to have you on this team doing great work on behalf of the animals and in the end for humans, because as we talked about, getting animals out of research labs, out of cages, and using human relevant methods, it's better for patients. It's better for the doctors and providers, giving them the tools they need to care for their patients. And it really improves the health overall. So it's a system that makes sense. We're trending in that direction. I'm so glad for the work that Emily and her team are doing. Please do check out her site if you're interested in learning more about the work they do.
Thank you so much for having me. It's a great conversation.
Been a pleasure, Emily. Thanks so much. If you enjoyed this episode or otherwise just interested in learning more, you can email me at john@johnliebermanmd.com. That's John, John, at JohnLiebermanMD.com. Thanks for listening. These conversations aren't always easy, but they're important and worth having. Take what resonates, ask questions, and keep learning. The content of this podcast is for informational and educational purposes only. It does not constitute medical advice nor legal advice, and it cannot replace professional consultation with your doctor, lawyer, or other licensed expert. Always seek guidance that's specific to your situation.